students, welcome to StudyZest. In today's class, the topics which will be discussed, these are the artificial propagation of plants, the three topics, the three processes, cuttings, layering and grafting. Then we'll discuss about the tissue culture, then how the tissue culture process is being done and what are the advantages of it. Okay. So before we start today's discussion, let's tell you something about us. As you can see, this is our YouTube channel, StudyZest Academy. You subscribe this channel, like our Facebook page, follow us on Instagram, join our Telegram channel. In this channel, you will get each and every kind of notification, each and every updates about the live classes and the study materials. So do join it. And you can also join through this WhatsApp community. The number is displayed over the screen. And for more updates, you can visit our website www.studygest.in. Now this is the interface of our app. It is freely available in Google Play Store. So you can directly go there. And this is a very user-friendly app. Go, hurry up and download the app, install it now. This is how our YouTube channel looks like. Do subscribe our channel and don't forget to hit the bell icon, so wherever any new videos will be uploaded in our channel, you will get instant notifications of that. And this is how our Facebook page looks like. Okay, so you like the page. Now let's start today's discussion. The first topic is artificial propagation of plants. Wait a minute. Okay. So what is it? Artificial. In the last class we have discussed about the natural method of vegetative propagation. Now this is the artificial one, which is the man-made. But how it is done, we'll discuss about that. The process, what is it? The process of growing many plants from one plant by man-made process or man-made method. This is known as artificial propagation of plants. Basically what is done here, the plants are grown from by the man-made method. Okay. Under this method, these three basic points are there. Regarding which we have to know. First is the cutting, second is the layering, and third one, very important, this is the grafting. Okay, so let's start with cutting first. This is the first method under the artificial propagation. In this picture you can see this is a plant, potted plant. Here by using a blade, a part of the plant is being cut, which is having its shoot part, the small stem, small fleshy stem along with its leaves. Okay, and that cut part is planted. How it is done? A small plant part which is removed by making a cut. Here the cut is being made with the help of this blade. So, or a sharp knife also can be used. This process is known as cutting. So the process by which or in which from a parent plant, a small part of plant is being cut either by using blade or a sharp knife. That process is called cutting. In this method, the cutting of the parent plant having some buds, maybe some floral buds are there, initial parts from which the flower starts to grow on it. It is taken and its lower part is buried in the moist soil. So as you can see what is being done, this is the cut part. Due to cutting, this is the cut part of plant. Alright. So this cut part of plant, this sand is there and the moist soil is there. This is planted in a new pot and from there new plant can originate. So after a few days we have to, in this state we have to keep this potted plant for a few days at least, and after a few days what we will observe that the cutting part will develop the roots. Initially the roots will be developed, and whenever the roots will absorb water from the soil, only then the food will be produced through photosynthesis, and whenever the food will be produced and it will be transported to each and every part of the plant, it will grow and properly develop. Then only we will find out the development. We will observe and notice the growth and development of other parts. So after a few days, the cutting part which we have already cut from the parent plant, that part will regenerate, that part will grow. The growth of roots will be initially observed, and it will gradually grow into a new plant. So what are the plants where this process of cutting is observed? There is rose, bougainvillea, chrysanthemum, grapes, sugarcane, bananas and cactus. These are the plants where by means of cutting, the artificial reproduction we can see. Now second is, after cutting, now let's come to layering. In this method what happens, a branch of the plant is pulled towards the ground and a part of it is covered with the moist soil, leaving the tip of the branch exposed above the ground. So what is being done, this is a parent plant there, this is a parent plant where one branch which is very near or close to the surface of the soil. This is the soil surface, and as you can see in this picture that this particular branch of this plant is present very near to the soil surface. So it is bent down and pulled inside the soil, deep inside the soil, and the part which is present inside the soil, that part is covered with moist soil. That part is covered with what? Moist soil. Okay. So whenever this moist soil will be covered, now let's see what we can see after that. After some times, new roots will develop from the part of the branch buried inside the soil. So the part of the plant or part of the stem or part of the branch which is present inside the soil, which has been pulled inside the soil, and it is covered with few layers of moist soil, that part will gradually show the development of small roots from there. Okay. Now whenever there will be formation of roots, after some time, after a few days, we'll see that there is growth of new roots. So whenever this condition will appear, then this particular branch will be cutting off from the parent plant, and the part of the branch which has developed the roots will grow and become a new plant. Alright. And very good example of this type of plant, this type of layering process is the jasmine plant. Next, we'll move on to what is grafting. So in this picture you can see that there is two plant parts, right? Two plant parts are there. One is written stalk, one is scion. So what is it? Let's discuss in detail. So grafting is a method in which the cut stem of two different plant parts, of two different plant origin, these are joined together in such a way that these two stems join and grow as a single plant. So this part and this part, their origin is not same. This is coming from one origin. One origin means from one plant, and this part, this stem part, is not a part of that plant. Okay, it is another plant. So the origin of this part is coming from the another plant. Okay, and gradually what will be done, these two parts will be joined and they will be forced. They will be forced or they will be made in such a way that that combination will give rise to a single new plant. This new plant will have the original characteristics of both the original plants. So the first plant is having a one set of characteristics, one set of characteristic features, right? So this plant will have one set of characteristics which may not be totally, which will not be, definitely will not be similar with the features of this plant. So the features of this plant and the features of this plant will be different. So the newly formed plant will show a combination of features. Some features will come from one plant and whereas some other features will be coming from the another plant. So that's why both the original plants will have partial expression of their carried characters in the new plant. So the cut stem of a plant having roots is called the stalk. So the part of the plant from which the root is taken, root is chosen, rather I would say root is chosen, root part is chosen, that is called the stalk. And it is the lower part, root is present in lower side of the plant, so it's quite obvious that it is the lower part of the plant because it has the roots. And the cut time or that part of another plant is called the scion, means the part of the plant from where the stem part is collected, the shoot is collected, shoot is chosen, that part is known as, that plant is known as the scion. And as it represents the shoot of the plant, so definitely it will be the upper part of the plant, unlike the stalk. Grafting is used to breed the fruits, trees and the flowering bushes like apple, peach, apricot and pear trees are often grafted. One very good example of grafting, it is very much it has been done in case of rose plant. In rose plant, what has been done earlier, from one plant the stalk is taken and from another part or another plant the scion or the shoot part is taken, and then these are undergone this process of grafting and gradually it has shown that it has successfully given rise to new rose plant through this process. Now let's see what are the advantages of grafting. Let me tell you that among these three artificial processes, artificial propagation, vegetative propagation processes, grafting is the most useful one and this is mostly accepted. Okay, whatever artificial propagation or whatever reproduction through artificial propagation has been successful, the most mostly the success rate has been found in this particular process of grafting. So this is very advantageous. So let's check out what are the advantages of it. Firstly, it enables us to combine the most desirable characteristics of the two plants in its flowers as well as fruits. What do you mean by desirable characteristics? Desirable characteristics are those which we desire. Okay, so wherever we can find a good characteristics of root of one plant and wherever we can find suppose good characteristics, good or desired which we want, that type of desired characteristics suppose we get in a plant from in its shoot part or the stem or leaves. Okay, so that part we can collect and they will be acting as an individual stalk and the scion, and then we can combine and we can see that wherever this grafting process is being possible to be successfully done or not. So in this way, if this is being successfully possible, then in the new plant all the good characters which we are desiring are known as desirable characters, those will be found together. Okay, some good character of root, suppose it's the good desirable character of root, the way it can be like the roots are very long and strong. So it can go very deep layers into the soil to absorb water. So it's a good quality. That quality suppose you want in a plant, so that from that part you can choose that, and for any good quality of the shoot, that desirable character, depending on that desirable character, the shoot of the plant can be chosen. And as you choose this, after choosing this, they will be combined, and their characters, the desirable characters, can be found together in the new plant in the form of flowers and fruits. By grafting method, a very young scion can be made to flower. Definitely young scion, maybe it is young for another plant, but whenever it will be grafted, whenever it will be utilized in the process of grafting, then it can give rise to a new flower and produce fruits quick fast when it is grafted to the stalk. So if the stalk is strong, then in that case any young scion also can be used to give rise to flowers and fruits. And this process can be used to produce varieties of seedless fruit. This is very important, because for the formation of seed we have to take the help of sexual reproduction in plants. But if suppose this is not possible, sexual reproduction is not possible, in that case, for the formation of seedless fruit, this grafting process is very, very essential. Here you can see this is the grafted plant. This is the grafted plant where the stalk and the scion, these are joined, and ultimately it gives rise to the plant, new plant. Okay, so these are the basic advantages of it. Now this slide is showing that how this, when in which cases basically this artificial vegetative reproduction or vegetative propagation is being used. Basically it is used usually for those cases or those plants which produce very few seeds. The plants which are producing very few seeds or do not produce any viable active seed. Okay, if they are not producing active seed, in that case they cannot undergo that, they cannot be involved, they cannot perform sexual reproduction. So in that case this artificial vegetative propagation is very, very essential for those. So in two cases, wherever they are producing suppose very few number of seeds, if these seeds somehow they are producing few number of seeds and they are not able to germinate properly, okay, so in that case that will be useless. And in that case also, where the seeds are not viable, seeds are not active, okay, in that case also this process is very, very essential. There this cutting, layering and grafting can be widely used, and the process can be going on. Next, coming to the next concept, that is the tissue culture. In these days this is very, very important process. As you can see in this picture that this is a rack, and in that rack many jars are there, this is one jar, and in this jar it's plantlet you can see, and in every jar small plantlets are there. So what is it? Tissue culture. We are culturing something in this process. So the production, what we are culturing? We are culturing new small plantlets here. Okay, whenever in the ground, in the field, in the agricultural fields, sometimes it is not possible. Okay, it may be dependent on the season, it may be depending on the availability. Suppose some plant which is very much needed, very much required, but that all the time it is not being possible, but its delivery, its requirement is very, very important. In that case what can happen? उस टाइम पे क्या हो सकता है, इस टिश्यू कल्चर की मदद से हम लोग जो है न्यू प्लांट्स बहुत ही ईजिली एंड थ्रू आउट द ईयर अवेलेबल रख सकते हैं, पा सकते हैं उसको. ओके, सो this is the process, this is the production of new plants from a small piece of plant tissue removed from the growing tips of a plant. The growing tips or the part, ये देखो ये बहुत important है क्योंकि इस process में एक small part, a small part of plant can give rise to whole plant body. Okay, so this small part of plant which is chosen for tissue culture, जैसे हम लोग दही जमाने के लिए थोड़ा सा दही तो हम लोग को चाहिए, तो वैसे ही ये प्लांट का एक ग्रोइंग टिप पार्ट है, जहाँ पर क्यों ग्रोइंग टिप ही क्यों ज़रूरी है? बिकॉज द ग्रोइंग टिप पार्ट्स ऑफ द प्लांट इट हैज़ द मेरिस्टेमेटिक कैपेसिटी, मेरिस्टेमेटिक एबिलिटी. मेरिस्टेमेटिक एबिलिटी मीन्स डिवाइडिंग कैपेसिटी. अगर डिवाइड नहीं होगा तो न्यू प्लांट कैसे बनेगा, आप लोग ही बताओ, है ना? तो ग्रो करने के लिए डिवाइड करना ज़रूरी है, और डिवाइड करने के लिए उस रीजन में, उस पार्ट में डिवाइडिंग सेल्स का प्रेजेंट होना उससे भी ज़्यादा ज़रूरी है ना? सबका एक कोऑपरेशन चाहिए, अदरवाइज दिस इज नॉट गोइंग टू बी पॉसिबल. ऑलराइट, सो द पार्ट ऑफ प्लांट विच इज चोजन फॉर स्टार्टिंग द डिविजन इन द टिश्यू कल्चर दैट इज कॉल्ड एक्स प्लांट. एक्स प्लांट हम लोग क्यों बोलते हैं? एक्स प्लांट मतलब बाहर से लाते हैं, बाहर से. ये हम लोग को जो रिक्वायरमेंट्स होता है ना प्रोसेस का, तो रिक्वायरमेंट्स होता है, हर एक प्रोसेस का होता है. वैसे इस प्रोसेस का जो रिक्वायरमेंट है फर्स्ट इज द एक्स प्लांट. ओके, सो ये एक्स प्लांट चाहिए, और एक्स प्लांट होना चाहिए ग्रोइंग टिप में. वेरेवर द प्लांट पार्ट द मैच्योर टिश्यू इज प्रेजेंट, दैट पार्ट इज नॉट डिजायर्ड. मैच्योर पार्ट लेंगे तो वहाँ मेरिस्टेमेटिक टिश्यूज नहीं रहेंगे, तो दैट पार्ट वी डोंट नीड. सो ऑफ अ प्लांट एंड इट डेफिनेटली ये हम लोग लैब में करेंगे. अगर लैब में हम लोग करेंगे तो वहाँ पर सॉइल नहीं होगा. विदाउट सॉइल देयर वोंट बी प्रेजेंस ऑफ वाटर मिनरल्स. सो दैट रिक्वायर मिनरल्स विच इज नीडेड फॉर द प्लांट्स, फॉर देयर ग्रोथ एंड डेवलपमेंट. इसके लिए हम लोग को क्या चाहिए? हम लोगों की सुटेबल ग्रोथ मीडियम मस्ट बी प्रोवाइडेड. तो इन द जार्स दिस मीडियम इज मस्ट बी प्रोवाइडेड, और दिस इज ऑल्सो कॉल्ड द कल्चर मीडियम. ये प्रोसेस को हम लोग टिश्यू कल्चर कहते हैं. अब ये कैसे करते हैं देख लेते हैं. फर्स्टली a small piece of plant tissue is taken from the growing point of the plant and is placed on a sterile jelly. The medium of the plant which is given, all the nutrition are present within the medium, that is present in the form of thick sterile jelly. Okay, sterile means without any contamination, कोई germ, कोई contamination नहीं है बाहर से. And it contains suitable nutrients and also the plant hormones. plant growth के लिए plant hormones चाहिए, plant growth regulators चाहिए, तो वो इस medium में present रहता है. alright, जो hormones है जो ये सेल को बनाने में हेल्प करता है, वो हॉर्मोन्स बहुत इंपॉर्टेंट है. ये रैपिड ग्रोथ के लिए बहुत ज़रूरी है. फर्स्टली जो ग्रो होके जो बनाता है, एक एक्सप्लांट से ग्रो होके जो फर्स्ट जो मास ऑफ सेल बनाता है उसको कहते हैं कैलस. एंड दिस कैलस is transferred to another jelly containing suitable plant hormone which stimulates the callus to develop roots. Okay, तो कैलस जो है वो एक इनिशियल ग्रोइंग पार्ट है जो अनकंट्रोल डिविजन से बनता है फ्रॉम द एक्स प्लांट, इनिशियल पार्ट. ओके, उसके बाद उसको वो जब ग्रो हो जाता है मतलब ऑल द मटेरियल्स, ऑल द nutrients inside the medium is totally used up, so it is transferred to new medium where newly available new set of minerals or nutrients, these are present in that new culture medium. Okay, next the callus with developed roots is put on a yet another jelly. वो क्या होता है, ये ट्रांसफ़र किया जाता है. ट्रांसफ़र करते करते क्या होता है, इसको ये whenever one small piece of tissue or plantlet is attaining its growth, how? By utilizing that nutrition, right? So whenever in that medium the nutrients will be finished, will be used up, so that medium is not needed at that time, it must be transferred to a new medium which stimulates the development of the roots. Definitely the hormones present in that medium will stimulate the development of the shoots. The callus having roots and shoots separate into tiny plantlets. Callus क्या होता है, धीरे धीरे रूट और शूट उसमें डेवलप करके अलग अलग रूट अलग शूट ऐसे नॉर्मल जैसे प्लांटलेट में होता है, सैपलिंग में जैसे होता है वैसे ये डेवलप करता है. एंड इन दिस वे मेनी टाइनी प्लांटलेट्स आर प्रोड्यूस्ड फ्रॉम जस्ट अ फ्यू ओरिजिनल प्लांट सेल और टिश्यू. ये आप देख रहे हो कि यहाँ पर एक कैरेट लिया है, वहाँ से एक छोटा सा सिर्फ एक्सप्लांट जैसे एक ट्रांसवर्स सेक्शन ऑफ रूट लिया गया था. उसके बाद उसको प्रॉपरली मीडियम में ट्रांसफ़र करते करते ये जो प्रोलिफरेटेड कैलस वो कैलस बना. इसके बाद और ज़्यादा ग्रो किया, और ज़्यादा ग्रो करने के बाद क्या होगा, इट मस्ट बी ट्रांसफर्ड टू अनदर न्यूट्रिएंट और प्रॉपर न्यूट्रिएंट कंटेनिंग मीडियम. कल्चर मीडियम बार बार चेंज करना पड़ता है. देन दिस इज फॉर्मिंग द एम्ब्रायनिक प्लांट and then ultimately it is transplant to agar medium culture. agar एक बहुत ही इंपॉर्टेंट मीडियम है जहाँ पर सारा न्यूट्रिशंस प्रेजेंट रहता है. तो यहाँ पर देखो, यहाँ से ये एम्ब्रायनिक पार्ट से, एम्ब्रायनिक प्लांट से कैसे एक न्यू प्लांट बन गया है. एक सिर्फ हम लोग स्लाइस लिए थे, वहाँ से न्यू प्लांट बन गया. यहाँ से विद इन फ्रॉम अ few slides it can produce the whole. So many plants, so many tiny plantlets can be produced. And the plantlets thus produced are transplanted into pots and the soil. इसके बाद इसको लेके हम लोग सॉइल में डाल सकते हैं और सॉइल में प्लांट करने से इट विल बी मैच्योर एनफ. राइट. सो अब देखते हैं कि इसका एडवांटेजेस क्या है. ये टिश्यू कल्चर प्रोसेस तो बहुत ही इंपॉर्टेंट है. क्या क्या एडवांटेज है? इट इज़ अ वेरी फास्ट टेक्निक, वेरी फास्ट टेक्निक जो फील्ड में रहता है. फील्ड में जैसे सीक्वेंशियली हमें सब कुछ करना होता है. फार्मर्स बेसिकली डू दैट, ओके, बट दैट प्रोसेस मे बी लेंदी क्योंकि वो नेचुरली होता है, वो बहुत सारे फैक्टर्स के ऊपर डिपेंड करता है. राइट. लेकिन यहाँ पर देखो ऐसा नहीं होता है. यहाँ पर जो हमें चाहिए वो हम लोग प्रोवाइड कर रहे हैं एंड दैट्स ऑल्सो आर्टिफिशियली. सो इट्स वेरी 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 गुड एंड वेरी फास्ट टेक्निक where thousands of plantlets can be produced in a few weeks time from a small amount of plant tissue. Okay, the new plants produced by tissue culture are disease free क्योंकि ये हम लोग लैब में कर रहे हैं. within the laboratory, within the sterilized condition this whole process is being conducted. So there will not be any chance of contamination. contamination free रहेगा. contamination free means disease free. So plants will be disease free and the tissue culture can grow the plants round the year. It doesn't depend on a proper time कि ये proper time जो winter में ही होगा ना summer में ही होगा. It doesn't depend on that. You have to just give the requirement and proper requirement. If the plantlet, if the callus tissue is getting the result accordingly you will get, irrespective of weather season. Even this whole throughout the year the tissue culture can be possible to grow new plants and very little space is needed. You don't need vast acres of agricultural fields and all. Only one lab is enough to produce thousands of plantlets. So space is also a factor. Very little space is needed here for developing new plants by the tissue culture. All right. So thank you for today. Next topics, rest of the topics of this chapter will be discussed in the further classes. Till then, thank you.